So good morning, everybody. Thank you for attending this workshop. So I will remind that FF4EuroHPC is a European initiative to support and to boost the use of HPC all around Europe, essentially by SMEs. So the first round of experiments selected during the FF4EuroHPC Open Call 1 are successfully closed now. So we organize 4 workshops to allow experiments partners present their realizations and the solutions they developed in the framework of FF4EuroHPC project. The first workshop includes experiments related to the medical pharma and the drug sector. 3 experiments will be presented as shown by the following agenda. So the first presentation will be done by Panagiotis Papadimitroulas and concerns HPC toward personalized medicine, the solution PediDose they developed for a pediatric dosimetry platform for clinical use by BIOMETEC and their partners. The second presentation will be done by Marino from Qubit Pharmaceutical and concerns advanced HPC-based drug discovery with converged deep physics and AI. And the last presentation will be done by Emiliano Costa and concerns Copernicus, the solution Copernicus they developed, which deals with a cloud-based HPC platform to support systematic pulmonary shunting procedures. And we will end this workshop by some conclusions. So a session of 5 minutes for questions and answers is planned at the end of each presentation. So please shake your hand if you want to ask questions or send your question by chat. I let now the floor to Panagiotis for the first presentation.
Hello, can you hear me?
Yes. And we see your slides.
Okay, and can you see in full screen my presentation?
Yes, almost. Yes, good.
Okay, perfect. So good morning, everyone. I'm pleased to participate in this workshop. Thank you, Samir, and the FF4EuroHPC for this final workshop that we are able to present the results of our experiments. My name is Panagiotis Papadimitroulas, and I'm the project director and co-founder of BIOMETEC. And today I will present the successfully closed experiment PediDose. It has to do with a pediatric dosimetry platform for clinical use, and it exploits HPC towards the personalized medicine. In this experiment, there were 3 partners, BIOMETEC, which is a domain expert, a Greek SME in the field of biotechnology, and coordinates this experiment. I know how, who is the end user of this software, and it is also a Greek SME in the field of medical software. And GRNet, our HPC experts, who are also partners from the Greek NCC. Also in this experiment, we had a subcontract on the IOTA HPC Center that provided us the HPC resources. So what is this experiment about, the PediDose? In nuclear medicine applications, we have the involvement of radioactivity. Ionizing radiation deposits energy in human body. It means that a patient that undergoes a nuclear medicine procedure, she or he takes a radiopharmaceutical, And then the radiopharmaceutical is concentrated in specific organs in the body. So then with the ionizing radiation, we have absorbed dose in several organs, and it can lead to cancer. Pediatric patients now are much more radiosensitive to the radiation than adults. The problem is that there is no way experimentally to measure this absorbed dose in each organ. It is called the internal dosimetry. And all the estimations until now are based on Monte Carlo simulations that is considered as ground truth. And of course, the problem is that we need to optimize the pediatric dosimetry protocol in order to consider the specific characteristics of each patient. Radiation dosimetry from radiopharmaceuticals has been a challenge for the scientific community for more than 50 years. And till now, there are no commercial solutions that incorporates the characteristics of patients, specifically in pediatric patients. As I told, there is no possibility to apply experimental dosimetry in clinical practice, and there is no possibility to validate the current dosimetry protocols in order to assess the absorbed dose per organ in the patients. And as far as Monte Carlo are considered the ground truth, and they can predict the dose, they can calculate the dose in each organ, they are so heavy to be implemented in daily practice, and it's not possible to have it in daily routine. What are Monte Carlo? Monte Carlo approach is, it is a statistical approach that solves deterministic problems and defines specified system using random numbers. Monte Carlo techniques were applied in nuclear medicine for many decades, and all these years there was an evolution of anthropomorphic computational models in order to be used in the Monte Carlo simulations to calculate the doses. Now the solution is that from an adult computational model, there are databases with the absorbed doses per organ, and the protocols to pediatric is done by rescaling these doses to smaller human bodies. So in PediDose, our objective was to develop a large simulated dosimetry database, which will lead to a new product, a clinical software product that will assist clinicians to have an estimation of the absorbed dose in such applications. This methodology is based on a previously developed methodology from another European project. In terms of our SMEs and the opportunity in industry, in BIOMETEC, we have already three preclinical imaging devices, so we are mainly on the hardware part of the market. And our aim is to extend and grow in the medical software, and this was a great opportunity to develop a new software for such application, as we had the possibility to exploit our expertise on this domain. And also we managed to have an autonomous software department within the company. For I know how, which is the end user, it enhances its medical software because they already have a clinical platform, and they are going to extend their applications beyond imaging and diagnosis to dosimetry. As I told, there are no solutions for personalized internal dosimetry, and using artificial intelligence with the developed database, we have a competitive advantage to the current dosimetry tools that are the ones that I described earlier. So the goal is to exploit advanced computational techniques. It means that we are using Monte Carlo simulations, advanced computational anthropomorphic models, pediatric models, and artificial intelligence techniques to develop a realistic simulated dosimetry database. The outcome is that we are going to have a decision support novel tool for the clinicians, and it will offer them the possibility to assess internal dosimetry and to optimize their pediatric dosimetry protocols. What we are using biodistribution of different radiopharmaceuticals derived from clinical data. We use the population of computational pediatric models. We had Monte Carlo simulations, of course, that executed on HPC, the creation of the simulated database, and then we had the training models based on regression machine learning techniques. With the aim to have the software afterwards to evaluate the product in clinical environment and commercialize it. In more details, why we needed the HPC. In our population, we had 28 pediatric models, and we covered the age range between 2 and 17 years old. And we used 5 radiopharmaceuticals. In order to have an accurate estimation of the dose, we simulated for each radiopharmaceutical 4 different time points of the biodistribution, and it concluded to almost 500 simulations. So in this experiment, we had the chance to have all the technology in our hands. It means that we had a very well validated tool, Monte Carlo tool, which is the GATE open source Monte Carlo toolkit, and it is quite well validated for dosimetry applications. We had the population of the models, the HPC resources that were provided through the FF4EuroHPC, and also the evolution of computing technology offered us the opportunity to apply machine learning prediction models. In our experiment, we had 4 different tasks. The first one was the standardization of the execution of Monte Carlo simulations in HPC and benchmarking them. Afterwards, we had the creation of the database, and in the task 3, we had the development of AI models and the creation of the GUI, the final software. And all these were running in parallel with the task 4, which was the management, dissemination, and exploitation task. What are the main achievements in the end of PediDose? We managed to standardize and benchmark the GATE Monte Carlo simulations in HPC, and they were validated with simulations that run in conventional CPUs. We had the creation of a large database, which is open and can be used by the community. Also, we developed predictive models to estimate, to predict the absorbed dose for each new patient that goes to a hospital and undergoes a nuclear medicine procedure. The accuracy that we managed is higher than 90%, which is more than acceptable in such applications, in dosimetry applications. We had the development of a GUI, and I know how managed to integrate the PediDose software in their clinical suite the Evorad software suite. And there were several dissemination activities and also exploitation activities in order to go to the market. An important activity was that 2 months ago we managed to participate in a national Greek program, which is the Symbiosis program, and I will tell you a little bit more later. So this is the final software that resulted from PediDose. Here you can see the platform, the GUI that the clinician is going to use, and the clinician here can put the characteristics of the patient, the anatomical characteristics, and can choose the gender, the age, the height, the weight, and then there is the possibility to put some more specific parameters of anatomical characteristics of his patient. And then the radiopharmaceutical is selected and the activity according to the protocol. If we calculate, we press the calculate button, we have the dosimetry, which gives the dose per organ for this specific patient, and there we can have a report for the clinician. Let me show a video that was produced by I Know How. It is a very fast demonstration of PediDose. This is the clinical suite of I Know How that is already installed in the most biggest hospitals in Greece. And here the clinician has the possibility to see the anatomical images of the patient. And I will fast forward a little bit. And there is the tools to measure specific parameters. Here, for example, the clinician measures the size of the lung. And in Evorad, here is the integration of PediDose, and if we click PediDose, then the software, the new software opens, and the clinician has the possibility to put the size of the lung of this patient here, and after the calculation, we have the prediction of the absorbed doses. The calculation is done in a few seconds, less than a minute, the prediction. And finally, by exporting a PDF, the clinician can have his final report. In terms of dissemination and exploitation activities, we had 3 presentations in different events, and there are videos on YouTube. We had the creation of this video by I Know How with the demonstration of the software. There are 2 conference presentations and 1 journal paper that is under review. It was just submitted. We had created a poster with the general concept of PediDose and the presence in several social media. In terms of exploitation, we had an initial market analysis and the SWOT analysis. I Know How managed to integrate PediDose to Evorad, and it is a great step for going to market. And regarding the symbiosis participation for PediDose, it is a Greek national program where hospitals are connected with SMEs and they can use advanced solutions that SMEs provides. And it is a great test point for us to test and evaluate PediDose in clinical environment, because we have the network with the hospitals. These are the 2 scientific abstracts that were presented in the 2 different conferences. The one has to do with the exploitation of HPC in order to execute the gate Monte Carlo dosimetry simulations, and the second one has to do with the development of the prediction models based on the simulated database. In terms of impact, we identified impacts in different sectors. First of all, there is a great social impact in the healthcare enhancement, as we are talking about personalized medicine in a very sensitive group of patients, the pediatric patients. Then we have the scientific impact because we are using very advanced computational techniques, and it goes beyond the state of the art in the dosimetry applications. And in terms of business and economic impact, we expect that this solution can provide SMEs with a very competitive advantage to this market, the medical software market, and it will strengthen the EU industry. I know how is going to extend their current software product, introducing the dosimetry features that I showed you in the video. And for BIOMETEC, we will manage to introduce our first medical software product beyond the hardware solution that we provide as a company. And of course, we expect new research collaborations between the partners. There were several benefits of FF4 to our experiment. There was the collaboration between two SMEs with a common goal to release a new product, and we are going to have a common marketing strategy. And of course, BIOMETEC managed to exploit its expertise as domain experts. We opened a new job position, which is standard in our company now, and we will manage to enter the medical software market. GRNet, which was our HPC experts, benefited by participating to a real business case, so they gained experience in interacting with business partners for commercial purposes. And I know how the end users of these experiments managed to enhance their customers, extending to the market of the dosimetry software. I would like, these are the collaborators in this experiment in order to execute the Monte Carlo simulations. And of course, I would like to thank the EuroHPC for this initiative and this opportunity. Thank you very much.
Thank you, Panagiotis, for this interesting presentation and this good job. Any question?
Yeah, that should be good.
Please. Okay.
So again, thank you very much. So today I'll introduce the results of this project that aim at accelerating and cutting down costs of drug discovery. If we can go to the next slide, please, Samir. So first, I'd like to give you a little bit of insights for those who are not experts in drug discovery. I think that's something you've heard many times, that developing drugs is a long and very costly process. It's very well defined, so there are many different stages before you can reach the market and give a drug to a patient. Basically, there is what we call discovery and development. Discovery is before the preclinical phases, when you start to test your compounds on animal beings and early humans. That's where you will, the aim of discovery is to identify one compound that will be suitable for further in vivo experimentation. At this stage, your aim is to filter, triage compounds and optimize them, starting from tens of millions of compounds. You only want to have the best compound for a certain given application. So it's I would say spread over several phases. The first one is, of course, identifying which target, which protein, or which RNA, what in the human body needs to be modulated, optimized, increased, or decreased in terms of activity in order to cure the disease. That's what we call a target identification, so target discovery here. And then once you know what is your target of interest, you are going to optimize compounds and identify compounds that are able to fit and to interact with this target. So the first thing that you do is triage a database of millions of compounds, and then reduce the number of compounds. So you start with tens of millions, sometimes even billions of compounds, and then this will go down to a few thousands, a few hundreds, and these hundreds of compounds are going to be modified to optimize their physico-chemical properties, toxicity, specificity, etc. That's what we call the hit to lead, the need optimization phase. That's what we wanted to optimize at Iktos and Qubit during this process. The reason why we wanted to optimize this phase is that it's still a half a billion phase and almost 4 years of studies before you can enter preclinical. So it's very long, and nobody can wait that long anymore in order to have novel drugs. One thing also that you have to keep in mind, pharma do go through on a regular basis. It's a cyclic basis. So let's say every 5 to 10 years, they undergo what is called a patent cliff. So due to patent expiration, their revenues are often at risk. So in the next few years, for instance, pharma revenues may disappear. So up to 40 to 60% of pharma revenues are at risk in the next few years. So they need to fuel novel pipelines, and they need to have access to novel compounds. And therefore, it's impossible to continue to have this long discovery process that take 4 or 5 years sometimes and are very prone to error. So if we can go on the next slide, please, Samir. So here I wanted to explain a little bit in detail. So that's the few phases, the initial phases that I showed just above. That's what really happened when we call target preparation, target to hit, hit to lead, and need optimization. For each stage, you need and you rely today on heavy wet lab experiments. So the first thing that you want to do is to have the crystal structure, the understanding of what is the target of interest. So basically, it's the 3D coordinates of all the atoms of your target. That's what we can see on the left side. Molecules and information on the environment, and if possible, you try to have some insights on dynamic interactions. That's pretty hard today because of the way we capture information on targets. So today you do crystal ray structure, mostly cryo-EM, and all these experiments need the protein to be stable, so not to move in space. Therefore, you lose all information about entropy, about flexibility of the proteins, and you need to do repeated experiments to grasp the different conformation a protein can take to identify pockets. Then today, in order to identify it, pharma do have developed libraries of compounds that they will screen through what's called high throughput screening. So robots having plates, and they are going to test it in real time. So if every time you need to change your assay, your test, it takes a couple of months. Of course, there are also some virtual screening approaches that allow to screen a virtual database of compounds. They are fast, of course, but it's a low resolution methods, and it's often it results in significant number of false positives and negatives. So what happens is, since you start with results of, I would say, poor quality and you cannot trust them, you enter a design, make, test, analyze phase, so the DMTA cycle, and not only one, but hundreds of them. So based on the initial results that you get from the target to hit phase, you are going to optimize the design of the compound, make them. So it's very common on that stage to synthesize 5,000, 10,000 compounds and test them one by one, see all their affinity, all their physico-chemical properties, toxicity, etc. Analyze your results and iterate again and again and again and again. So it's very labor-intensive process. And today, the issue is that simulation and AI tools are not, the ones that are available are not as good as the experiments, so you cannot trust them, and you build your models based on experimental data. So you still rely on a lot of experiments. And that's what we try to tackle. That's what we tackle today with Iktos on this project. So if we can go on the next slide, please. So this project had two objectives. The first one was to develop a blended called deep physics and AI pipeline, aiming at accelerating the drug discovery. So developing a pipeline that is as good as the experiment and that will allow to reduce the number of DMTA cycles and optimize all the compounds in silico and validate them with simulation, and then successfully apply the pipeline to a target that resists drug discovery over the years. It's called USP7. So if we go on the next slide, please. So this pipeline has been developed by gathering the strength of each company. Iktos is a company that is very well known for its generative modeling algorithms. So they are aware, they are able, based on constraints that we send, to optimize a compound in order to meet several objectives. And that's complex because very often the properties that you are trying to reach do compete with each other. So optimizing property A is sometimes and very often decreasing property B. So you need to do multi-parametric optimization and be able to explore a chemical space. But then the issue is that what AI is very good at generating, it cannot validate your compound. So you need a method, a tool to be able to validate in silico. Elsewise, instead of synthesizing 5,000 compounds as on average, you end up in synthesizing 50,000 because with AI, you are much more creative than with human being. And that's where we enter. With our engine, we are able to simulate all compounds and accurately predict interactions between molecules. So this will guide the design and synthesis of all the compounds. So here, there is one thing also to keep in mind. One of the issue and one of the challenges that we had to face is managing frequencies. So generating novel compounds with AI is very fast. We're talking about a couple of minutes. Validating compound with high-level physics and measuring all interactions with exquisite precision is done in matters of hours. So one of the challenges that we had, it's a very old challenge, is how to manage a time difference between two different bodies and how to design a pipeline that would benefit from HPC in order to have physics that drives AI and AI that iterates thanks to real evidence and real information. So if we can go to the next slide, please. And then there is a reason also. The one thing that we had to choose is a target, because developing a pipeline and applying to something that is meaningless is easy, but we wanted to work on a target that has a real sense for the pharma industry. USP7 is a very well-known target. Been years that pharma is trying to develop compounds targeting USP7, because USP7 plays a pivotal role in many different cancers. So it's on the pathway of a lot of cancers, and most of them are still cancers that resist most of the treatments. Breast, such as some breast cancers, colon cancer, you still have, chances to die with these cancers is still quite high. And that's why USP7 is a good target for the pharma industry. Yet the issue is that since there was little success in the world regarding this target, there is little data, and the data available is of poor quality. Also, there is a few, and the understanding of the target engagement, so how it will interact with the rest of the body, is also poorly known. So that's why here, physics, high-level physics, gives a lot of insights, because we cannot rely on data. Therefore, if you don't have data, you cannot train models, etc. So each partner brought his tools and assets in order to tackle this challenge and develop an innovative pipeline. We can go on the next slide, please. And that's how we developed the pipeline. So basically, what we try to do is to mimic entirely and optimize the classic wet lab-driven drug discovery pipeline and optimize it entirely on HPC infrastructure. So the first thing we did was, instead of performing hundreds of experiments and getting access to the crystal structure of a protein, we simulated entirely the protein over a very long biological time. and its evolution. So since we have a high resolution physics, we are able to really grasp what the protein undergoes in terms of conformational changes. And for this, we need to do well, it's optimization and a lot of sampling procedures that requires using several hundreds of GPU in parallel over a very long time, so several days and even weeks sometimes. Based on that information, we are able to know where to target the protein. So what are the most probable pockets in terms of existence? So protein, since the protein is going to move and change conformation, sometimes there are pockets that disappear if you only do one crystal structure, but overall, in average, over time, they do exist. And these are the ones that are interesting. So that's what we tried, finding information on these pockets and then addressing them. And then based on that, Iktos could develop its proprietary generative algorithms in order to generate a lot of small molecules that would fit perfectly in that pocket and match all the criteria that we were expecting. Once these molecules were done, we did validate them by computation, so simulation. And one of the key criteria that was measured was the affinity and interaction between what we call interaction maps, so between the small compound and the protein. And then in the end, it's only while a classic full drug discovery cycles would have sent thousands of compounds to synthesis, we only retain a few tens that are currently synthesized and tested. So if we go on the next slide, please. Here I try to show you how we did impact and how we did redesign if we put both in parallel. So task one, as I mentioned, we did replace and it's really a very powerful tool for target preparation and target understanding, especially engagement. Task 2, 3, and 4 led by Iktos were the ones that we do what we find, hit and then optimize them. And what's very important is the it seems a small detail, but what we did, the interconnection between task 4 and 5. So if we go on the next slide, I'd like to dig a little bit deeper into this interconnection. The design, make, test, and analyze cycle is very long because doing a calculation is very easy. I mean, you can do it in a matter of days or even hours or even minutes. What is always stopping or increasing the timeline is when you need to synthesize a compound, because I mean, real world and the real synthesis is never that easy. So this can take months, especially if you are trying to develop novel chemistry. And basically, when you tackle a target that has been known for years, and by years, I mean 20, 30 years, you have to go through novel chemistry because all the classic chemistry, all the well-known chemistry has been tested. And therefore, you need to develop a novel pipeline. And that was only with computation and with the fact that we had access to very high-level physics fast, so thanks to HPC, and novel generative algorithms, that we were able to break the cycles. So instead of having these DMTA cycles, we had the DT cycles, or even DA, so design analysis very quickly. And that was the very challenging part. So intertwining algorithms that don't have the same pace, don't have the same outcome, don't run on the same infrastructure was quite challenging, but we succeeded in it. And that's the very novel and innovative workflow that is one of the outcome of the project. So if we go on the next slide. Also, just to give you a couple of numbers, as I mentioned, we do use a lot of physics, complex one, that requires using a lot of GPUs. So overall, on the project at Qubit site, we did burn about 150,000 GPU hours. And the same in CPU hours. So there was a lot of jobs that were submitted. And some jobs were very big, using tens, hundreds of GPU in order to gain a lot of insights, information on the target. But it paid. If we go on the next slide and the outcome, the results are very, very let's say satisfying, and we are very happy with the project. The first one is it's shown on the graph on the upper right of the solution, upper right of the slide, sorry, we developed a solution that is as accurate as experiment. So the graph shows the comparison between experimental measurement and calculation. And what we're able to do is to have computation that are as good as experiment on complex target. So we do have a pipeline that really mimics reality, that really have digital twins of what happens on assays, and we know we can completely rely on the pipeline. Then on the bottom right of the slide, Iktos also developed a very good triage system that, given about 1 million of compounds, was able to reduce the number of compounds we needed to validate to about 30. Because of course you don't want to use very high-end methods to thousands of compounds if they are not needed. So we've both optimized our workflows in order first to quickly and accurately fuel AI pipelines with good and curated information. And the second one, an AI pipeline that was able to triage a lot of compounds in order to only select the best ones for physical validation. And that's already, I mean, developing this pipeline is already a very good outcome of the project. The second one is that with this collaboration, we are very happy. Unfortunately, we cannot show you the structure, but we have found several good starting points for further lead optimization in a very short amount of time. So developing the pipeline was the initial part of the project. It took some time, and then we applied it really to discovery. And now we have chemical families, chemical starting point that are novel and interesting. We both scaled up our infrastructure on HPC, especially thanks to GenC in France. So we worked a lot with them, and that's how we are able to use that many GPU hours. So thanks a lot to GenC for the help. We gained also novel insights on other methodologies. For instance, Iktos has developed a pipeline using what is called the MMGPSA. So that's really I mean, major outcomes for us because in a very limited amount of time, thanks to the project, we're able to implement and scale methodologies to an extent that was not feasible before. If we go on the next slide, please. So what are we going to do now with this project? First, we will continue the drug discovery project. So we aim at further investing about 2 million euros together in order to bring these compounds to a state where they are what we call pharma materials. So we can engage with pharma in order to out-license the compounds. This will lead to further patent applications, publications, of course. You are probably aware that the pharma world is quite a secret world. So we wait as long as possible before sharing information outside. So in the pharma, so very often you don't patent before you are at stage phase 2 clinical trials. That's not something we are going to do. So we'll patent later. But if we patent too early, then there is no market value anymore because the expiration of the patent will occur before the drug is on the market. So it's important to know when to patent and how to optimize it. We know within the time that we had, we could not completely optimize the pipeline. So we know there are still optimization possible. That's something we are working on. And of course, continuing the optimization of the compound. That's why it will require the further 2 million investment. Also, this pipeline works. We know it can be applied to complex targets, hence to easy ones also. Although there is no easy target in pharma, everything is complex and tough. But we are very happy that we can now offer this solution to third party, and it will be available beginning next year. So there will be business development led by Iktos mostly and us into proposing this pipeline to third parties. So yeah, if we can go to the next slide. So just to sum up, drug discovery is a quite complex process where you need to optimize a lot of different properties. And today, due to limited access to computing power or to restrained physics, simplified physics, people had to cope with uncertainties. And if we go on the next slide, just to conclude, and that's where HPC has a major impact, because the more the physics, the more the computing power you have, the better the accuracy and the resolution that you can dig into. and therefore, the better the drug optimization process. You cut cost, you cut time, and you can deliver novel drugs faster.
From Italy, thank you for joining today. I'm going to present the experiment, the results of Copernicus, the experiment 1006, the extended title. Let me move. Okay, is a cloud-based HPC platform to support systemic pulmonary shunting procedures. Okay, I work for RINA. RINA is an Italian engineering company, and we are the coordinator of the experiment. Okay, this is the outline. There are many topics, so we grouped it in 4 items, basically: the introduction, the development of the activities during the experiment, the final considerations, and then the conclusions. So first of all, I will just introduce the consortium. In silico trials is SME, which will benefit at first of the outcomes of the project. It is an industrial end user. And then there is we are in total 5 partners. The second one is RBF Morph, which is a technology expert and independent software vendor. Then there is Fondazione Toscana Gabriele Monasterio, the application expert, the clinical end user. And then there is, of course, RINA Consulting and as a technology expert and coordinator. And then Sineca, which is the HPC Center of Italy. And they act as HPC expert and host center. Which is the problem, okay? We'll talk about congenital heart diseases that are roughly 1/3 of all congenital birth defects and the 7th cause of death in children. Okay, so this is the problem. Okay, so without the ability to modify the prevalence of congenital heart disease, intervention must be aimed at improve survival and quality of life. Okay, so there is a specific operation called the modified Blalock-Townsend shunt, MBTS, that is a palliative operation on cyanotic heart diseases, but it consists basically, can you see this item? There is basically the intervention for to have this implantation called the shunt to try to balance the systemic pulmonary flux, basically. But this operation is associated with significant mortality. Okay, why? Because the position, the length, and the diameter of this element, the shunt, is up to the surgeon, basically. So the surgeon don't have surgeons today a powerful means to support in the decision apart from the CT scans. Okay, so the idea of the experiment is to provide a powerful support system through medical digital twin. Okay, so the Copernicus application aims to generate this to support the surgery planning at best when critical conditions occur, typically. Which is the target is since there are typical critical conditions, the need is to provide this medical digital twin, in particular the computational fluid dynamics ROM, reducer order model of the district of the patient within 2 days, basically 48 hours. Okay, on the right you will see the workflow, okay, of the Copernicus application. Basically, the surgeons provides medical images that are segmented to generate the CAD model of the patient. Then the shunt is positioned on the CAD model in the baseline configuration. Computational fluid dynamics case is generated in which the geometry of the shunt is parameterized according to using the radial basis function mesh morphing. Then this parametric computational fluid dynamics model is run on HPC to generate lots of variants considering the different shapes of the shunt, and then the results are processed to generate the reduced order model. The medical digital twin can be so visualized and evaluated in real time in order to get the fluid dynamic behavior of the district in function of the shape of the shunt. And so the surgeon is capable to evaluate if critical conditions from a fluid dynamic point of view occurs or not. Okay. This is more in depth the workflow. In the middle, we can see the 3 phases of the Copernicus application. So the pre-processing, the computing, and the post-processing. So in each of these, we can see the images that allows that basically easy the comprehension. We have the CT images and the segmentation on the top left. So there is the CAD generation of the district, then the shunt is added to the CAD model. Then the computational fluid, the mesh is generated, and the CFD case of the district is generated. The shunt is parameterized through mesh morphing techniques, and this information goes to the HPC, in which we compute the variance through a design of experiment study to generate the reduced order model and to finally visualize the medical digital twin, basically, that allows the surgeon to modify the shape of the shunt in function of the shape modifiers defined through mesh morphing, and evaluate real time which is the modification of the fluid flow in the medical district. So this is one of the main achievements we gained during the project. So the Copernicus called it the Copernicus numerical case scenario for MBTS. This consists basically on a specific framework that guarantees an accurate reduced order model of the district with a valuable that is valuable from medical point of view. Okay, so in particular, it is composed by 4 items. Okay, so first of all, an high fidelity CFD case that was generated performing a sensitivity analysis on the mesh. Then a set of suitable shape modifiers were generated, in particular 12 in our case, and these modifications have a medical significance. Okay, then we set up a design of experiment suitable to generate an accurate ROM. In our case, 150 design points were used. And we also identified through a scalability test, basically, the computing process needed of the CFD solver to generate a good analysis in a reasonable time. Now we'll better detail what we did on HPC. Okay, the main IT developments on HPC are 2, basically. First of all, we set up the HPC environment of Cineca. In particular, we used the Galileo 100 cluster, okay, hosted by Cineca. and here we install the software, the licenses. We set up all environmental variables, libraries to run all software needed for computing. In particular, the computational fluid dynamics solver, the Morpher tool, and the ROM builder preprocessor. Then the second, this is one of the added values of our project, is the implementation of a specific procedure that allows to use more than one single computing node of the cluster. And this is, as we will show later, this allows to boost a lot the computing, and so that the computing time is decreased a lot. Okay, basically a set of snapshots run in parallel on different computing nodes using the Slurm workload manager. Okay, some results. This is the basic result with the single node strategy. So in this case, we use a single computing node of Galileo, and automatically we run in sequential way 150 design points. And here on the bottom, there is the time in hour for the pre-processing, computing, and post-processing phase of Copernicus. We see that the whole workflow lasts here 22 hours, so we are just below the requirements of 48 hours. But we can see on the right through a pie diagram that the computing phase covers basically the 74% of the whole procedure. So we can improve the whole workflow working on HPC, and we did this. Okay, we show here. So adopting the multi-node strategy, we run, as I already introduced, a set of variants on different nodes. Okay, since we had in our license pool 5 licenses of the CFD solver and in infinite licenses of the Morpher tool, we used 3 and 5 licenses of the CFD solver that run in parallel. And on the bottom, you can see the results similar to what I already presented. The results when 3 and 4 licenses are used. Okay, you can see that basically the computing time is decreased a lot, till covering for 5 licenses 1/3 of the whole procedure. Okay, and in this successive slide we compare on the bottom the single node diagram, the multi-node with 3 nodes, and the multi-node approach with 5 nodes. Okay, and you can see that how the part of the relative to related to the computing stage is decreased a lot, and in the multi-node with 5 nodes case, we are able to run the computing phase in just 3 hours approximately. So the complete workflow is covered in 9 hours. So this is a very good results we gained that showcase that the advantages provided by the massive use and the massive use of HPC computing. Okay, in this slide we summarized some of the main results achieved. So we succeeded in integrating implementing integrated and effective numerical procedure to generate the reducer order model of the patient-specific vascular district, in which the shunt is parameterized through mesh morphing. We set up the Copernicus numerical scenario, so a framework which guarantees to generate an accurate and valuable medical digital twin. Okay, so we demonstrated the competitive advantage of the use of a massive HPC. We generated also a business model and exploitation plan. We performed more than three actually, but the three main dissemination activity. We presented at EuroHPC Summit in Paris, the experiment. We also presented it in the World Biomechanics Congress in Taipei. And we also published an open access journal, MDPI, the work done in the first phase. Then we got also an increase of knowledge, basically, on the application of radial basis function mesh morphing in the medical sector. Increased the knowledge of the capabilities of the medical digital twin. And we also assess a new HPC cloud-based service in the medical sector. This is the exploitation roadmap we drafted during the proposal. We are here in September. Okay, we succeeded in the completion of the project with good results in our opinion, and now we are ready to do some actions, okay, some initiatives. The first of which that is very important is to try to use in the research field the application, to improve the application, to get and try to get the certification, to then exploit from the economical point of view the use of this application. But first of all, we have to get the certification. Here we summarized some of the business benefits of the Copernicus application. The in silico trials, this SME will reach new clients after the certification, offering services per clinical use. Fondazione Monasterio will save basically money, reducing hospitalization, basically. The ISV RBF Morph expect an increase of their business due to the fact they can sell the services and licenses of the Morpher tool. We also expect an overall increase in 4 years due to the increase of our offering in the medical sector. And Seneca, of course, we can increase their business as a HPC supplier for this specific type of application. From societal point of view, we think that we will be able to lower the incidence of post-surgery complications, recurrence, decrease the days of hospitalization. We estimated, and all the figures are reported in the final report, and we are able today to guarantee a fast, reliable response. So we are able to generate the medical digital twin of the patient-specific medical district to support surgery in less than 48 hours. Actually, today we are able to provide this in a time below 10 hours. That can be also improved, in our opinion. So some lessons learned. We got honestly a deep knowledge on the use of commercial tools adopted for running Copernicus application. Basically, the software are based on the ANSI suite, apart from in-house tool to reconstruct the images that is based on neural networks. And we also there is also we got the comprehension of the crucial importance that an effective use and an effective implementation of a good strategy for HPC can help a lot. It can drastically decrease the computing time. Next step. We can, of course, the possible idea is to generate a web application to allow surgeon to evaluate via web the medical digital twin on their own. Okay, we can make less user dependent the procedures, trying to boost to automate some of the processes and the actions needed in the pre-processing phase mostly. We can further disseminate the implemented procedure. We have to search for collaboration outside the consortium with stakeholders in the medical digital twin, in the biomedical field. And we have for sure, before business, to consider the process to get a certification in the medical field, which is a not easy task for sure. And in conclusion, we described the implementation of the Copernicus application, detailing the problem to take all the procedure and the numerical means used to accomplish the work. We showcased the strategic advantage of the HPC usage. We also outlined the economic and societal advantages of the use of the application we developed. And we also described the roadmap and the rationale of the exploitation we outlined. Okay, so I have finished my presentation. I take the occasion to thank FF4EuroHPC board and project for giving us the possibility to develop this application. We think that we did a good work, and we hope to collaborate again in the next future.